monkeys eat bananas. Got a lot more to cover, but not a lot of time to do it. So I'm going to pick up the pace a little bit, and you guys will have a little bit more responsibility put on your shoulders to read through the chapter and kind of fill in a lot of the blanks. Okay, I'm just going to kind of hit some highlights from here on out with you. So let's draw this back down. See where we're going next. Got a couple videos to take a look at and just a few more concepts to check out. All right, so blood, we discussed this right here. Okay, difference between serum and plasma, why serum is preferred. Serum is the result from allowing the blood to clot, as we mentioned. This term right here, hematopoiesis. Poiesis is the making of. Hema, of course, refers to blood. Hematopoiesis is the making of blood cells, all blood cells, red blood cells, white blood cells, and so on. Hematopoietic stem cells, these are the stem cells in which all blood-related cells are derived from. Okay. It just depends on the path. A stem cell is a cell that can make any kind of cell. Hematopoietic stem cell is a stem cell that can make any kind of blood cell. This picture is out of your book, and let me tell you what I'd like you to know about this picture. First off, we can just see everything begins with one hematopoietic stem cell. It just depends on the path that it takes. Okay. Now, what I'd like you to know, you don't have to memorize all these steps in between. That's just kind of a waste of studying, in my opinion, but what we do need to know is what's going on down here at the bottom. Okay, what these results are. So here, we can see if they take one path, you'll end up with red blood cells. We know the function of red blood cells to transport these blood gases, so oxygen, CO2. Okay, great. There's another cell called a megakaryocyte. That'd be important to know. Okay, megakaryocyte. Mega means large. What does kary mean? K-A-R-Y. What's the difference between a eukaryote and a prokaryote? Nucleus.
Yeah.
Okay, so megakaryocyte. So you see a large nucleus in this large cell, and basically it gets fragmented into like 1,000 pieces. And those little tiny pieces are what's known as platelets. Okay, so a platelet is not a whole cell; it's a fragment of a very large megakaryocyte. Of course, we know that they help with clotting. And then your white blood cells, here you can see your 5 types. Okay, so you have your 3 granular cytes, neutrophils, basophils, eosinophils, and your 2 agranular cytes. So your monocytes, and then your lymphocytes, even though they showed the 2 lymphocytes, but lymphocytes. Looking at the agranular cytes again, okay, we can see the little granules present. We can see what they do. We should really already summed all this up. So you need to know the function of each of these white blood cells, which we basically covered on the board. Monocytes, lymphocytes, you need to know their function too. They're specific. Okay, lymphocytes are, monocytes are in the blood. They can migrate into the tissues and then become macrophages. As we can see here, very, very large. So you're going to ingest and kill. They can also develop into those dendritic cells that we saw in the earlier picture, okay, which are very similar to macrophages. They help to break down antigens, kind of kickstart an immune response. And then we have natural killer cells, which I did not put on the board. Natural killer cells are beneficial to attacking viruses and cancer cells. They are not specific. Non-specific derivative of your white blood cells. Let's do a concept check. first one, true false. Lymph is propelled through the body by the action of smooth muscles.
No.
That's false. What's wrong with that sentence?
Skeletal muscle.
Skeletal muscle, yep. All right, the blank functions to process worn out red blood cells and also helps to filter pathogens from the blood. Spleen, yep, that is the spleen. And then you should be able to describe basic function of the white blood cells. Not just these four, but all of them, of course. All right, we need to talk about these processes. Okay, so I'm going to be kind of hopping around a little bit and just kind of summarizing some of this for you, some of the main points. I would like you guys for sure to read up on a lot of this. We're going to start with phagocytosis, which again is the ingestion and killing. You need to be attracted to the organism first, to the pathogen, and then it needs to be able to engulf it and needs to be able to destroy it. It's not as easy as it sounds because some of the pathogens can have anti-phagocytic factors, as we've talked about before. Neutrophils do a really good job at ingesting and destroying. Again, these are your first responders when you're looking at inflammation, when you're looking at infection. They show up pretty fast. High neutrophil count, typically present when you look at early stages of an infection. Very high numbers. Your three main phagocytes, neutrophils, monocytes, and macrophages. Okay, we have those on the board. Okay, which active phagocytes, these macrophages and these monocytes, along with the neutrophils. Here's one more thing that we can kind of base this what I have up here, this side over here. So here we have monocytes in the bloodstream, right? And then they can migrate into a tissue and they're known as macrophages. So we got that covered. Now there's some tissues in the body that always have macrophages present and they stay in that tissue only. When that happens, they are known as histiocytes because the term histo means tissue. So histiocytes are macrophages that stay in a particular tissue. Don't go anywhere else. They stay in that tissue. If we look at this picture, this just shows your monocytes in the blood, which we covered, macrophage in the tissue. Great. Monocyte in the blood, macrophage in the tissue. Histiocytes are macrophages that stay in one tissue only. There's three examples up here. Okay, so within the lungs, you have what's known as alveolar cells. They stay in the lungs. They're histiocytes. They're macrophages in the lungs. Kupffer cells are found in the liver. Langerhans cells are found in the skin. They are resident macrophages, also known as histiocytes. That's it. That's all that we're looking at. So permanent residence, that's where they stay. I'd like you to know the events that take place with phagocytosis. I'm going to sum this up for you by looking at this picture. I would not say step 1 is this, step 2 is this. Just kind of know the flow of it. And you guys already know this. Here's the bacteria. Phagocytic cell, your white blood cell, gets attracted to it in some manner, sees the PAMPs, ingests it, releases chemicals that kill it, and then the digested debris is released. I mean, that's the summary of phagocytosis. There is a fancy table, a figure that shows you these steps, but we already are familiar with it. Chemotaxis by the phagocyte. So here you're looking at like a neutrophil. It gets attracted to the bacteria. Bacteria attach. Okay. Recognizes these little PAMPs as foreign. So it engulfs the bacteria. Here they are engulfed. They're engulfed in a unit called a phagosome. They're engulfed. Lysosomes within the cell that contain digestive enzymes fuse with it. It has a fancy name. It was a phagosome. merge with the lysosome. Now they call it a phagolysosome. Just kind of combine the two terms. Little enzymes kill the bacteria, and then you just have the debris. And that's what takes place. Okay, different enzymes. Okay, peroxidase, proteases, DNAs to break down the DNA, RNAs to break down the RNA. There's some lysozyme in there. So they're going to destroy the bacteria. Little video clip.
Microorganisms that cause disease are called pathogens. When pathogens enter the body, white blood cells called phagocytes pass through the walls of the blood vessels and into the surrounding tissue. There they can ingest the pathogens and pathogens' toxins, absorbing and digesting them. Phagocytes can also destroy pathogens by releasing enzymes. Having absorbed a pathogen, the phagocytes may also send out chemical messages to call in other white blood cells.
Okay, so just a basic look at phagocytosis. Next up, we got inflammation. So we'll take a look at what happens with inflammation and then leading to fever. So this is kind of a cool picture that I just grabbed off the internet that takes a look at the 5 characteristic signs, symptoms of inflammation. So first one is heat. So you can see this flame is getting close to this guy. So some heat. Here you see some redness. Okay, so face red, just embarrassed, lost his little cloth here. Swelling, you can see here in the belly. Some pain. Okay, so stub the toe, experiencing some pain. And then potentially loss of function. So here you're seeing limbs falling off, so loss of function. Those are your 5 characteristic signs that you see with inflammation, signs and symptoms. Rubor, redness, tumor, swelling, dolor, pain, calor, heat. Okay, so definitely know the terms for inflammation. You don't have to worry about the Latin terms for it. It doesn't help. I mean, it helps to be familiar with them in case you hear them later, but I definitely want you to know the characteristic signs and symptoms, which we can see right here. And anybody can initiate inflammation. So if I were to just kind of scratch, my hand, take my nail and scratch my forearm right here, I can initiate inflammation. So when I did that, it's like it's all warm and tingly. So I feel the heat, right? Eventually, you can see a little red line up my forearm. Okay, so you can see the redness. I had the warmth, the redness, the swelling, kind of depending on how bad the inflammation is. You can actually feel a little bit of swelling. Okay, right where I scratched my arm. If I stimulated some of those nerve endings, it can actually hurt. I didn't do it hard enough to hurt. I'm not going to purposely hurt myself, as an example, but if I did that hard enough, yeah, that can cause some pain. And depending on how severe the injury is, you may lose function. So this example right here, you can see there's a puncture to the skin, puncture to the finger. Well, then you might lose functioning of that finger. Maybe you don't want to type on a computer with that finger if you just punctured it. Okay, so that's possible. But all these stages work together just to kind of amp up your immune response. Okay, so let your body know something has happened. So here we see the characteristic signs and symptoms resulting from this injection of that thorn. Stages, really nice picture in your book that runs through the various stages. So we can take a look and then recap with the video. So here we can see, here's initial damage. So this nail gets driven into some tissue through the skin. There's bacteria on that nail, so they're dropped off within the skin. You can see there's some damage to blood vessels within the body. There's some mast cells releasing some chemicals, which is going to attract more immune cells. Vasoconstriction happens immediately, which kind of backs up the blood, which is going to help to promote clotting. And then it's going to, you're going to have some vasodilation happening to get more blood and immune cells to that location. So that's immediate. And then here we can see, due to the constriction and dilation, we can form that clot. Neutrophils, they show up right away. They show up pretty quick, and they're going to start engulfing and breaking down the these bacterial cells. Here we can see how they're seeping out of the blood vessels. There's a term for that. It's called diapedesis. You'll have that on in your notes in a couple of slides. So they seep out. It's called diapedesis, transmigration, or emigration. There's like 3 names for it. We see all that going on. Okay, here we see the pus forming. So it's like broken down bacteria, cellular components, okay, a lot of debris from the infection. And then when all is said and done, these super large macrophages show up and they help to finish killing and cleaning everything up. Okay, so quick look at inflammation, and then we have a video to show you as well in just a moment. Okay, this just kind of recaps a lot of the chemicals which are at play. Edema is just a swelling which is going to take place as a result of this inflammation, okay, which is going to consist of the pus and debris and dead bacteria and so on. Diapedesis, transmigration, emigration, 3 terms for the ability of the white blood cell to seep out of the blood vessels. Pyogen means to generate pus. Pyrogen would be to generate heat, but pyogen is pus-generating bacteria. Some bacterial infections result in just more pus because the bacteria like to generate or help to lead to the formation of pus, like Streptococcus pyogenes. It's actually named for the fact that it's a pyogen. And we know macrophages clean up at the end. So here we can see the diapedesis, again, transmigration, emigration, seeping out of the blood vessel into the tissue space to undergo this immune reaction. So we'll start this video. There's some weird music in the beginning, but it'll end in a few seconds. I don't know why it's there. But it's a really good video that you should probably watch numerous times just to see everything that happens with inflammation.
Inflammation is the protective reaction of vascularized tissue to local injury. Acute inflammation is the early, almost immediate reaction of tissue to the injury, and it is the first phase of wound healing. Acute inflammation can be triggered by cell or tissue damage, or by the presence of dead cells or noxious agents such as bacteria. Acute inflammation typically occurs before the immune response becomes established and aims primarily at removing the injury-causing agent and limiting the extent of tissue damage. Acute inflammation occurs in 2 overlapping stages, vascular and cellular. In the vascular stage, arterioles and venules near the site of injury constrict briefly, then dilate. Dilation promotes congestion, while an accompanying increase in capillary permeability leads to the movement of fluid into the affected tissue, resulting in the 5 classic signs of inflammation: redness, swelling, heat, pain, and loss of function. As fluid leaves the capillaries, the blood remaining in circulation becomes more viscous, it flows more slowly, and clotting occurs. The cellular stage of acute inflammation is initiated by the movement of phagocytic white blood cells, or leukocytes, into the area of injury. The leukocytes begin to adhere to the vessel wall, and then, in a process called emigration, squeeze through the wall and move into the inflamed tissue. The leukocytes wander through the tissue, guided by chemical signals in a process called chemotaxis. The cellular stage culminates in the leukocytes engulfing and degrading the bacteria and cellular debris in a process called phagocytosis. Products of phagocytosis, along with plasma and blood cells, form exudates, which accumulate, causing swelling and pain. Exudates are composed of serous fluid, red blood cells, fibrinogen, or tissue debris, and white blood cell breakdown products. Concurrent with the events of the vascular and cellular stages, chemical mediators release bioactive agents that act to mediate the inflammatory response. Mediators are derived from the cells or from plasma. One of the first mediators of an inflammatory response is a cell-derived mediator, histamine, found in high concentrations in the mast cells of connective tissues adjacent to blood vessels, as well as in blood basophils and platelets. Histamine is released in response to a variety of stimuli and causes dilation and increased permeability of capillaries. Serotonin, another cell-derived mediator, performs similar actions. The three major plasma-derived mediators are present in the plasma in precursor forms that must be activated, usually by a series of proteolytic enzymes. The kinins increase capillary permeability and stimulate pain receptors. The clotting system traps exudates, microorganisms, and foreign bodies. The complement cascade causes vasodilation, promotes leukocyte chemotaxis, and augments phagocytosis. Acute inflammation comprises a variety of complex protective responses to injury that together lay the groundwork for the next stages of the body's recovery: immune response and tissue repair.
Okay, so pretty good video that sums up a lot of the portions of inflammation. Quick glance. I'm doing a quick glance at fever. Let you guys know what interferon is. Let you know what complement is. We'll do a concept check, and that's really all we can do today. We're very close to the end of time. So as far as fever is concerned, when you take a look at fever, you see the hypothalamus in the brain is reset to a higher temperature, so your body needs to catch up. Okay, so you're going to have some chills set in because your muscles are going to contract, try and get your blood temperature to match what your brain is saying the temperature needs to be. So that's going to take place. And if you think about any benefits to fever, one benefit would be, well, the temperature is higher, bacteria are not going to like that. Okay, so you want that temperature higher to prevent bacteria from growing. Okay, so that's definitely a benefit to fever. There's another big benefit to fever where your metabolic reactions, a lot of your immune reactions, will happen faster at a higher temperature. So it speeds up your body's response because the temperature is higher. A third benefit to fever would be some of your white blood cells give off iron. Okay, they give off some iron and other nutrients that the bacteria like to eat. They actually like to feed off of those products. Well, when your body's temperature gets higher, your white blood cells don't give off those nutrients. They don't give off the iron. Then the bacteria don't have those nutrients to feed off of. So those three benefits are discussed here. So be familiar with the benefits of fever. So should we always try and reduce a fever, or can they be beneficial? They can definitely be beneficial. Okay. So pyrogens are the causative agents of fever. It can come from your body, it can come from the bacteria. Interferon. If we look at what interferon is, I will tell you guys right now, interferon most commonly is something produced in response to a viral infection. Is interferon produced in response to a bacterial infection? Sometimes, but it's very common with viral infections. A lot of antiviral medications, they're types of interferon, because this is how they work. I can sum up interferon, and then I can use you guys in the class as an analogy to how interferon works. So first off, the way interferon works, let's look at this picture, is this particular cell, this host cell, is hit with a virus, right? Okay, so this host cell is eventually going to die, okay, because of this viral infection. What the host cell does in response, yes, it's going to be destroyed because it is infected with the virus, but what it does is it produces interferon. Okay, and then it takes this protein and it releases it. This interferon protein travels to neighboring cells. They receive this protein, and the protein's like a little message saying, all right, start making some antiviral proteins because the virus is coming your way. So then this cell and the other neighboring cells, they're ready. So when the virus gets to them, they have antiviral proteins ready, and they're able to combat the virus. Okay, so it's kind of like a warning signal.
Mm-hmm.
The analogy I like to give is I can kind of call on you guys. Let's say Megan gets to class early. She's the first one to show up, right? She comes to class, and I'm like, all right, guess what, Megan? Here's a pop quiz. She's like, what? You know, pop quiz. She's not ready for it. Let's say she doesn't do very well. This first cell getting hit with the virus, not doing well, okay, that's like Megan, right? But she's like, well, I don't want my classmates to bomb this pop quiz. So she takes out her phone, and she texts Kaylee and texts Anna, and lets them know, hey, when you guys get to class, there's a quiz. So they're gonna look over the material, and they're gonna walk into class, and they're gonna be ready. So I hit them with this pop quiz, but it's not gonna really do much because, you know, they're ready for it. Okay, so it's kind of like that warning signal, like Megan sending out a message. So this first cell is sending out a message to the neighboring cells, just kind of warning them, having them get ready. Okay, so that's interferon. Last thing we have today is complement, and then a concept check. So with complement, it's a series, a couple dozen proteins in the blood. Okay, they're going to set up what's known as the complement cascade. It's just another method that your body uses to destroy bacteria. It's a whole cascade of events. You guys don't have to memorize the steps. I can show them to you. They're kind of crazy. Okay, there's a number of steps going on here. You have one complex bind, and then you have your C4 and your C2 and your C3A and B. They're all going to bind. It gets a little crazy. So we can sum it up. And basically what happens is you're going to have your initial complement proteins bind, which is going to attract other complement proteins. When all is said and done, you have complement protein number 9. Okay, so your C9s, your complement protein 9s, end up forming a ring. This ring is called a membrane attack complex, the MAC. This little ring punctures a hole in the bacteria. Bacteria can't maintain its contents, it's lysed, the bacteria dies. Okay, so the complement is a bunch of blood proteins working together to ultimately puncture holes in the bacteria, which the bacteria cannot survive. So that's what we see with complement. It's done as a complement to other immune reactions and infections. It's like the sole thing that happens. Okay, so in this picture down here, you can see that the C9s form this MAC, this membrane attack complex, punctures a hole, bacteria is lysed, bacteria dies. That's what complement is. Final concept check. Okay, so you should be able to describe the steps of phagocytosis. Okay, so the bacteria gets attracted, ingests, destroys, uses those enzymes from the lysosome, destroys via bacteria. What are the 5 symptoms of inflammation? Heat, redness, swelling, pain, loss of function. Okay, those are the 5 signs and symptoms of inflammation. All right, blank is the tendency of white blood cells to migrate in response to a chemical stimulus.
Chemotaxis.
It's actually not diaphoresis, it is chemotaxis. Okay, so the attraction of bacteria due to chemicals is chemotaxis. Diaphoresis is the migration of the white blood cell out of the blood into a tissue space. Okay, so it leaves the blood vessel, enters into the tissue space. True or false? Fever should always be treated.
False.
False. Interferon is produced in response to an infection, most commonly by what?
Virus.
A virus. Yes.